Good morning, Suzuki Science. This is Mr. Ellis here on week 3, and we are going to look at genetics. We are still looking at genetics. Last week, we looked at some of the DNA replication and how DNA is able to produce itself. One of the things we did not go over is what happens if a mistake occurs, but there's going to be gaps in your learning, so just be aware. This morning, we uploaded a Sanger sequencing biotech technique, and it's a part of the biotech stream of things that we'd like you to learn about. You're not going to learn as much as we'd like, clearly, and that will be another gap that'll be in your learning. And also this morning there was a interactive workbook that you could have worked through to get a feel for what this lesson's about. So now I'm going to teach it, and I'm going to skip a number of things I don't consider all that important in terms of getting a better understanding of the overall process. So once the DNA replicates itself, that's a great process because that's necessary for mitosis and every single time the cell's replicating. We also have another major process, is the other big piece of learning, another big idea, and that is something called the central dogma of molecular biology. The fact that DNA can be transcribed into RNA that can be translated into proteins. When you're working through this lesson, so with the grade 10s and the grade 11s, they have these Google worksheets, and I've highlighted in green what they're supposed to know, and they can kind of minimize it in 3 hours maybe. With you kind of just pick and choose. You have a midterm mark, you have it going off to the university shortly, and then anything you do above and beyond that in terms of our evaluation can only complement. So in terms of the work, you basically do as much or as little as you deem necessary in order to get an understanding of the material and demonstrate your understanding in these weird circumstances. All right, so I don't have a Google sheet that's made for you, but we will have objectives that you should do your best to understand. So, the central dogma of molecular biology. You know already that I like to start off my lessons with some kind of engage, something interesting that's related to our course material. And I want you to take a couple minutes at least, it's a 4-minute video, and explore the world of dark DNA. Some of you have talked about dark matter, and you've talked about dark energy, and so it only seems fitting that biology has its own version of the dark side. So in order to get you connected, you can watch the Seeker video. Some of you are probably familiar with this online school of scientific thought, and he does a really good job of engaging you as well. So listen to him and what dark DNA is all about.
Dark matter is one of...
All right. So next, if we were in class, I'd spend a whole stream of thought on the various important scientists that we need to know for genetics and for some of the other units. In this case, I have spent a little bit of time in the last section on replication, but not as much as I would like. And because of our timing and because of what's important, knowing who the scientists are and what they've contributed is not really that important for our learning. So on this slide, you'll see a link, and I keep on providing these links to you. I encourage you to go to this link, and this link is exploring Beadle and Tatum. You'll notice that Beadle and Tatum are in red, so they're the ones that are important. And what Beadle and Tatum did, if you click on that link, you're going to come to this site here. I really like this site. It's an old site. It's called DNA from the Beginning, DNA FTB. And they do a really good job of connecting the classical genetics from grade 11 and the molecular genetics of grade 12. And so they have... it's always in the same pattern. They have the concept that's in a paragraph. They have the animation that you can click on and work your way through. It's not voice, it's just text and pictures. And then they have some other things to support that particular concept. So as I look at what Beadle and Tatum had provided, if you watch this little animation, they'll step you through the scientific process that they went through. And what's always amazing, what I find amazing about these scientists is that they're using, I don't want to say primitive equipment, but they're not using stuff that we have here in 2000 to 2020. They're using some pretty traditional chemical techniques. So please explore that. And if you go to that link, you'll see that they summarize through some pretty nifty sciencing using mold, bread mold. They were able to determine that the mold required some very specific amino acids. And if they were able to affect a specific enzyme responsible for the metabolism of the amino acid, the mold would not grow. Okay? So They went through a very deliberate scientific method, and they were able to determine that a mutation to a single gene was then able to impact the enzyme. And from there, they developed this idea of one gene directs one enzyme. And from that, we eventually learned that it's not just an enzyme that's affected, it's actually a protein. So I would encourage you to take a little bit of time to explore Beadle and Tatum. They're the ones that allowed us to come to the understanding that one gene equals one protein. This is actually not true. Remember, in biology, especially in high school, we teach you a lot of foundational stuff, and then as you learn more, we teach you other things that kind of go against what you learned. Okay? But for now, we learn that one gene codes for one protein. Okay? And this led to something called the central dogma of molecular bio. In the central dogma of molecular bio, we understand that DNA is a genetic information. It can be transcribed into RNA, and that can be translated into protein. Okay? So those 5 simple words is represented by courses and courses and courses of information. We are going to capture it in a week. Okay? Now, what's interesting is DNA creates protein, but it is not direct. The analogy that we often use is the analogy of a page here. The analogy of a work site. So let's say you have a work site and you want to build a building. So to build that building, you have these blueprints. Okay, so you got these blueprints, and they have the structure of the building inside of it. That's my castle. And so you go to the work site and you got all the work equipment. All right, you got this work equipment here, bulldozer. Okay, don't bother me. I can't draw. Bulldozer, BD. And you have other things, people working, lots of equipment. And if you bring the blueprints over to the work site, what's going to happen to the blueprints? There's a good chance that the blueprints that they come to the work site, they're going to become destroyed. Okay? So instead of bringing the original blueprints to the work site, what we do is we often will make a copy. Right? And so that copy will be hopefully exact, because you don't want any mistakes. And then you can bring the copy over to the work site. And the copy can allow you to build the actual building for real. Right? And then while the building's being built, if your instructions, your copy here, becomes a little, torn, frayed, or destroyed, it's not that big a deal. Why? Because you can go back to the original and make a new copy and bring the copy to the work site. And this is what happens in our cell. The DNA codes for the protein. The protein is the end product of a lot of things that we want to create inside our cells. And so instead of taking the DNA out to the violent, very disruptive area of the cytoplasm, a copy is made, the RNA, which is brought into the cytoplasm, which allows the protein to be constructed. So there are a lot of scientific experiments and a lot of history that shows how this whole happens. I've just summarized it in 2 minutes. RNA we talked about in the first unit, in biochemistry. So you should have an understanding of biochemistry and RNA. We know that RNA and DNA are very similar. The differences include that DNA has deoxyribose and RNA has ribose. So it's a slightly different sugar. It has 1 extra hydroxyl, 1 extra oxygen. We know that DNA is typically double-stranded, whereas an RNA is single-stranded. You can find examples of double-stranded RNA, but generally speaking, it is a single strand. We know that DNA includes the nitrogen bases of A, C, T, and G. We know that RNA does not have thymine, instead it has uracil. If you look at the chemical structure, which we did during biochemistry, they are very similar. We know that DNA is the informational code that codes for everything. RNA is a messenger that goes between DNA and protein, and it also has quite a few other roles. In this course, I'm going to specifically talk about mRNA, tRNA, and rRNA. Now, one of the links you'll see is this here from Learn Genetics. Learn Genetics is another excellent site, and they have a lot of material that's perfectly designed for the high school student. And if you play through this little animation here, you'll get a feel for how DNA and RNA differ from each other. Okay, so I encourage you to play with that. Also, if we go to the site, the DNA from the beginning, we can see Mr. Crick from Francis Crick and James Watson. He is going to talk about how they determined the RNA was the transitional molecule between DNA and protein. So it wasn't just automatic, it took some experimentation. And then finally here on another website, another link that you'll see on the PowerPoint is this Learn Genetics interactive animation. And on here you can see the original DNA that's inside the nucleus and how it's going to be transcribed to an mRNA and how that's going to leave the nucleus and come out to the cytoplasm and then be translated into a protein. The help of the tRNA and the rRNA and the mRNA, those three RNAs together are going to create a protein which will then be, whatever the end job is. Okay? So that brings us to the gene. So with the gene, we are trying to relate DNA, which has 4 letters, to proteins of which there's 20 amino acids that compose protein. So there's got to be some direct relationship between the DNA's 4 and the protein's 20 amino acids. So what's that relationship? Could it be a one-to-one relationship? Could we have an A, let's go black here. Could we have an A, a T, a C, and a G that's going to code for 20 different amino acids? Clearly not, because if there's a code of 1 letter, we can only code for 4 different amino acids. So, 1 letter, that's not going to work. Okay, suppose we go with 2 letters. So we've got AA, AT, AC, and AG. If I do all these combinations with 2 letters, what's the maximum number of combinations you can have? Well, we do a little bit of math magic in class. I'll be kind of looking at you, you'll be kind of looking at me, and I'll be hoping that your math skills will be equal to the task. It might be first thing in the morning, you'll be not answering me, and eventually I would say, okay, there's 16 possible combinations. Okay, so knowing that there's 16 possible combinations and that there are 20 amino acids, could a 2-letter code be the genetic code? And the answer, once again, would be no, there's not enough. So that brings us to 3. So at 3, you might have, AAA, and AAT, and AAC, and AAG, and all these other combinations. Let's see if we can see a pattern here. There's 4 possible combinations here, so that's 4 to the 1. There's 16 possible combinations here, which is 4 squared. There are 3 choices here of the 4. So if I go 4 cubed, this will represent a final number of 64. There are 64 combinations if we use a 3-letter code. And as a matter of fact, the genetic code is 3 letters. Okay? So this genetic code of the 4 DNA, the 4 DNA letters, they will be transcribed and have a relevant RNA code. And those RNA codes represent something called a codon. A codon codes for a specific amino acid. So every single triplet represents an amino acid or a stop, meaning don't transcribe anything, okay? Or don't translate anything, just stop. So when we go to our code here, this table represents the genetic code. So you should be able to read this genetic code. So let's kind of explore how we go about reading the genetic code in case you are not able to see it immediately. So here I have the genetic code table, and let's say we have a code sequence that says AUG, and I want to know what amino acid that represents. So I go to the first base, A, I go to the second base, U, and I go to the third base, which is G. And so that ends up being methionine. Suppose I gave you a code of C, U, G. All right, so let's take a look at this one. I got C as my first base, I got U as my second, and I have G as my third. So that's going to be leucine. Let's try one more. Let's say I want to go with G, A, C. So I go to G, I go to the A column, so G and A, and I look at C. And I see that's aspartic acid. Now what you'll notice with the genetic code that almost every single amino acid has more than one code, except for methionine. But if you look at phenylalanine, it has two. If I look at leucine, it has 6 different codes. Isoleucine, it has 3 different codes. Glycine has 2 different codes. So all these things here, many of them have more than one code. And so what that does, it actually makes the genetic code a little, we call it degenerate. That means there's more than one code for each amino acid, which means a little bit of wiggle room. So let's say we have a mutation, and the mutation changes a CUG, which was coding for leucine, to a CUU. So what would happen? Let's take a look. We know CUG stands for leucine. CUU also stands for leucine as well. That means that when I take a look at this, that if there were a mutation that caused one base pair to shift, it's not that big a deal in this case, because I'd still have leucine, the amino acid being produced, and if that leucine was necessary for the protein to work properly, we'd be all fine. So that's okay. I also want you to notice that there are 3 very special sequences. They are UAA, UAG, and UGA. And so all 3 of these represent a stop. And we'll explore what that means in just a moment. Okay? So if you come across these 3, everything stops. This is the genetic code, and this is how you read the genetic code. In addition to the mRNA, which is your classic RNA that you're going to see, there's other types of RNA as well that are important. And these other RNA include the transfer RNA, which carries an amino acid from the nucleoplasm to the, from the cytoplasm to the binding site, where they're going to create a growing polypeptide of amino acids. So the tRNA has a very distinctive shape. We'll look at that in just a moment. And we have these ribosomes, which is the factory where the proteins are created, and that's also our RNA. So let's practice our translation here. And we're going to practice it for the first time. Let me just refresh this page. So I encourage you to play with this as well. So this is a DNA strand. The DNA strand opens up, and then you're going to write down the complement. So what is a complement for A? It is going to be U. Complement for T is A, and so on, right? Right? So now we've transcribed the DNA to an RNA. Now you'll notice that in addition to the stop codons, there's actually another codon that was on that page, and you didn't see it highlighted, but it's called the start codon. And the start codon is a codon that where translation will always start, and it's known as the AUG. So I'm going to find AUG. AUG stands for MET, but it's also start codon. It's always start, always start. So now that I have found the start, I'm going to grab my amino acids, CUA, CUA, GAC, GAC, GUG, GUG, UUC, UUC, phenylalanine, and then UAG, UAG. It's stop. And there we go. We've now transcribed and translated the that last process was translation, the DNA strand into a protein. That's the process. I would encourage you to watch this video. This is a great TEDx video. This is a group of people. Drew Barry's the main person here, but they've demonstrated what the molecular machinery looks like inside the cell. And it is a faithful rendition of what's happening in. If you take a look at this, if you haven't seen this before, I strongly encourage you to take the time, sit back and relax, and watch it for a period of time, because it's really amazing what's happening inside the cell. So that represents the central dogma, and it's the first part of our lesson. I'm going to break the lesson into chunks, and so the next part of this chunk is going to be transcription, and then the last part will be translation.